Hi everyone, today I'm going to walk you through the Carbohydrates chapter for AQA A Level Biology, which is part of the Biological Molecules unit. Now, I'm sure that the content in this video will definitely cross over with other examples, as the content is quite similar in a lot of ways. Also, at the end of the video, I'll be going through a few exam style questions and their mark schemes, as I know from experience that the biology mark schemes are the absolute worst. So, let's make a start. In carbohydrates, the single unit, or the monomer, is called a monosaccharide. I'll just draw a kind of circle to represent one unit. Now, there are three monosaccharides that you need to know about: glucose, galactose, and fructose. Now, the unique thing about glucose is that it exists in two forms, alpha and beta glucose. Now, you're probably wondering, they look the same, but in actual fact they're not. In alpha glucose, this OH group is, as you can see by the diagram, below the ring. And with beta glucose, this hydroxyl is above the ring. Now, the way I used to remember this was it's the opposite way to what you think. So you would naturally think, oh, alpha above, beta below, but it's actually the other way around. And the significance of this will become more important later in the video. Now, let's talk about the condensation of monosaccharides. Recall from the first part of the unit that condensation is the joining of molecules to form a chemical bond excluding water. Condensation of two monosaccharides produces what we call a disaccharide, and many monosaccharides produces a polysaccharide. Now, the bond that is formed between the monomers is called a Glycosidic Bond, and that term is really important as the examiners really like it when you use that term. Also, sometimes you will be expected to draw the Glycosidic Bond when the question gives you the two monomers, so it's kind of like this. And also, it is very important that you write plus H2O, or else you sometimes don't get the mark. Now there are three disaccharides that the biology AQA specification requires you to know: maltose, sucrose, and lactose. So I'll just write malt for simplicity. Malt, so it's suc. I apologise for my very messy writing. Now, maltose is the easiest one to remember, as it is made of two glucose molecules. Sucrose is made of glucose and, wait, my bad, fructose isn't a disaccharide. Ignore that. Apologise. Glucose and fructose. And the other one that you need to know is actually lactose. So lac. Now, lactose is made up of glucose and a sugar that you probably haven't heard of before called galactose. Just like galac for simplicity again. Now, sometimes exam questions ask you about the enzymes that catalyse the reactions that break down, for example, lactose to glucose and galactose. It's very easy to remember. So the enzyme that breaks down lactose is called lactase, sucrose is sucrase, and maltose is maltase. It's very easy to remember. So the next part of this chapter is to know the structure and function of the polysaccharides starch, glycogen, and cellulose. So we'll start with starch. Starch is mainly found in plant cells. Starch is a polymer of alpha glucose, which is very important to remember as it often comes up in exam questions. Now, starch has what we call a helical structure, which is kind of like a coil, but examiners sometimes don't accept the term coil, so it's best to use the term helical. Now, because starch is helical, that means it is very compact. I'll just rub that out. Pen. That's very most, but you can kind of read that as compact. Now, this means that it can be easily hydrolyzed to alpha glucose for uses in respiration. And one other thing to note about starch is that it is insoluble. This means that it can't affect the water potential. I'll just cross WP to represent water potential, which is very important in plants as plants need to regulate their water content so that they can grow efficiently. Now, the next thing you need to know about starch is you need to know how to test for it. These tests are very important as in my exam there was actually a massive 5-marker about these tests. So the test for starch is you add iodine or potassium iodide, doesn't matter which one you put, to your sample that you're testing for starch, and if there is starch present then the sample will turn black, as you can see by this image. So this black colour, what we call a precipitate, shows that there is starch present, and this yellowy colour shows that there isn't starch present. So next we'll talk about glycogen. Glycogen is also a polymer of alpha glucose. However, what is different about glycogen from starch is that it is highly branched. As you can see, it branches off from here. Now, this is a very important adaptation because that means it can be readily broken down to glucose for uses in respiration, so it enables glycogen to act as a storage molecule in animal cells. And this is why glycogen gets its name animal starch, as it is present in animal cells. So finally we'll go on to cellulose. Cellulose is different from starch and glycogen in that it is a polymer of beta glucose. Now this is where the significance of alpha and beta glucose is clearly shown, because as you can see, the molecules of beta glucose are inverted. So this group is on the top here, it's on the bottom here, it's on the top here. And then you can see that the glycosidic bonds, so the hydroxyls, are inverted as well. Now you're probably thinking, well, why is it inverted? Well, because of the arrangement of the atoms in beta glucose, they must adopt this configuration so that glycosidic bond formation is easier. Now, the interesting thing about cellulose is that the hydroxyls can form hydrogen bonds between other molecules or other chains of cellulose, as cellulose comes in chains. These lines just represent the hydrogen bonds. These hydrogen bonds form tough fibres. These tough fibres are called fibrils. Now, these fibrils are what give cellulose what's called a high tensile strength, which gives it its supporting role in providing strength and rigidity to plant cell walls, as you can probably recall from GCSE. Also, cellulose has straight chains, which is important in hydrolysis to beta-glucose, again for use in respiration. Now, the last thing you need to know from this chapter is the Benedict's test for reducing and non-reducing sugars. A reducing sugar is a sugar that can act as a reducing agent and can be oxidized. Remember from GCSE the oil ring mnemonic: oxidation is loss, reduction is gain. This means that itself loses electrons, but it makes other molecules gain electrons. Now, the specification doesn't make you remember this, so it's not important to remember for your exam, as there will never be a question on it, but it's good to remember. Examples of reducing sugars are your monosaccharides that we have mentioned, such as glucose, galactose and fructose, and an example of a non-reducing sugar would be sucrose. Now, to test for a reducing sugar, we do what's called a Benedict's test. So if we have a test tube with your sample in it, and we add what's called Benedict's reagent, it's BR just for simplicity, and Benedict's reagent is blue. Now what we do now is we heat it. You don't need to know what temperature or how long for. If you get an exam question about Benedict's test, the mark scheme just says heat with Benedict's reagent. You don't need to know any specifics. Now, if we have a reducing sugar, the result would give you a brick red precipitate. I know this colour isn't brick red, but I don't think I have a brick red pen. So that is a positive test for reducing sugar, so there is a reducing sugar present. Now if it's negative, then the solution will remain blue. Now you need to know how to test for non-reducing sugars. And what we do is you have your sample, and what we add in this time is hydrochloric acid. And now what we do is we don't just heat it, we boil it. The purpose of the boiling is to make sure that the non-reducing sugar that you're testing for is hydrolyzed. Now, after it's cooled, what we do is we need to add an alkali to neutralize the solution, as it is very acidic and the test wouldn't work if it was acidic. So we add an alkali such as sodium hydrogen carbonate to the solution. Now, after we've added the alkali, what we do is we perform Benedict's test again, exactly the same as we did before. And now the presence of a brick red precipitate shows that we have a non-reducing sugar. That's it for the content. Now we can get on to some exam questions. Now, oh no. The examiners love to ask you about these kind of bead contraptions. So let's read the question. Cow's milk contains the sugar lactose, so we can highlight that. Many cats are unable to digest cow's milk because they are lactose intolerant. Cow's milk can be made suitable for these cats by treating it with the enzyme lactase to hydrolyze lactose. This makes the cow's milk lactose-free. Beads are coated with lactase, that's a pretty important point, and placed in a tube, as shown in the diagram below. Cow's milk flows over the beads and the lactose is hydrolyzed. So we can. Attaching lactase to the bead is a more efficient use of lactase than adding lactase directly to cow's milk. Suggests 3 reasons why it is more efficient to attach lactase to the beads. Now, the first thing when tackling exam questions is you need to look at the type of question you're answering. So for this question, it is a suggest question. Now, suggest questions often don't require knowledge from the specification. They require your own ideas, kind of like an application of your knowledge. And also, we need to highlight that you need 3 reasons. Now, one thing about these bead contraptions is that you can reuse the beads. So that is an important point to make. Also, the enzyme is more stable because obviously in the tube there's a more controlled environment, so you haven't got as many changes in things such as temperature and pH. Also, another point you can make is that it allows a continuous process, so nothing runs out. So if we check the mark scheme, accept 3 suitable suggestions. And notice down here it says 3 max. That means if you suggest any more than 3 of these, then you don't get extra marks or anything. You can only get 3 maximum. So this is the point that I mentioned before. Lactase beads can be reused. They accept lactase and the beads are not wasted, but less lactase used is insufficient, so that wouldn't get the mark. No need to remove from milk, that's another acceptable point. Allows continuous process, that is what we put. And the enzyme is more stable. And to avoid end product inhibition. You don't actually learn about end product inhibition or feedback inhibition at A level, so you're probably unlikely to write that. So the next exam question is this one. Monosaccharides and disaccharides taste sweet. The lactose-free milk made after hydrolysis of lactase tastes sweeter than cow's milk containing lactose. Now we need to highlight the important points in the question. And the important point here is after hydrolysis of lactase. Now we need to figure out why this is sweeter than just lactose, using our own knowledge applied to the question, as it is a suggest question. Now the most obvious thing is that lactose would be hydrolyzed to glucose and galactose as lactose is present, as I mentioned earlier in the video. Now, as glucose and galactose are 2 separate sugars and lactose is just 1 sugar, there are more sugar molecules present, so that would make the solution more sweet. So let's check the mark scheme. Lactose hydrolyzed to galactose and glucose, which is what we put, so there are more sugar molecules. Now, here it highlights the word more, so you need to include that somewhere in your answer to get the mark, so you can't say there are lots of sugar molecules. It's comparative, so you need to say the word more. And then there's this idea, so more / different receptors are stimulated / sugars produced are sweeter. But you're more likely to write somewhere along the lines of this. So the next question is give. So it's stating facts. 2 ways in which the structure of starch is similar to cellulose. Now the first thing to note is that they're both polysaccharides, as I mentioned earlier. And next is that they are linked by glycosidic bonds. Again, the examiners really like you to use the terms that link the monomers together, such as glycosidic bonds, peptide bonds, ester bonds, and so on. Next question is another give question. This time it's 2 ways in which the structure of starch is different from cellulose. First thing to note that they like to hear is that starch is a polymer of alpha glucose and cellulose is a polymer of beta glucose. Now note that it is not asking you to compare, so you don't need to write starch is a polymer of alpha glucose but cellulose is a polymer of beta glucose. You don't need to do that, just waste time. Also, starch is coiled/helical. The examiners much prefer you to write helical, so I'd write helical here instead of coiled. So here's the mark scheme for part AI. So they're both polysaccharides, and another point you can make is that they contain the elements glucose, hydrogen, carbon, oxygen. Now I've written here that glycosidic bonds are included. Another point you could also make is that they have 1-4 links, which means the glycosidic bonds are made between carbons 1 and carbons 4. And also, starch, I forgot to mention, has hydrogen bonding within the structure. However, cellulose also has hydrogen bonding within separate molecules. But they actually ignore that, so you don't get the mark if you write that. And also remember, it's 2 marks maximum, so you don't get extra marks if you write more than 2 of these. Now the next question. Obviously, starch contains alpha glucose. And here it says assume it refers to starch. Try to avoid using the term it whenever you're answering something, and make it really clear that you're referring to what you're writing about. So if you're writing it and the examiner thinks you're referring to, say, cellulose, it won't give you the mark. Here it says accept converse arguments. So an example would be if you say starch is a polymer of alpha glucose whilst cellulose is a polymer of beta glucose, but only if linked directly to cellulose. So if you don't make obvious that you're comparing it directly to cellulose, you won't get the mark. And also they accept forms alpha glycosidic bonds, but I wouldn't recommend that you write that. And yes, they accept helical and coiled. And you can also write this compact, branched or not straight. One thing I did forget to mention earlier was that there is actually a branched form of starch called amylopectin. Amylopectin. I apologise for my terrible writing on here. I'm not used to writing on a computer. And something else to point out is that the helical form is called amylose. And then starch contains 1 to 6 bonds as well. Glucose and monomers are the same way up, as we said earlier that in cellulose the glucoses are inverted to allow the easier formation of the glycosidic bonds. And also there are no hydrogen bonds between the molecules, as there are in cellulose. And also they don't form fibrils, as the fibrils are only formed when there are hydrogen bonds between the molecules. And again, 2 marks maximum, so you don't get more marks if you write more than 2 of these. Now I think this is the last one we're going to go through. Starch and cellulose are 2 important polysaccharides. The following diagram shows part of a starch molecule and part of a cellulose molecule. So this is starch, this is cellulose. Explain the difference in the structure of the starch molecule and the cellulose molecule shown in the diagram above. Now, as you can see from the question, it is an explain question. And this means that you have to explain why something is happening or how something is happening. You don't just write what happens, because you won't get all the marks. Now, the first thing to point out is something obvious that we always mention. Starch is a polymer of alpha glucose, while cellulose is a polymer of beta glucose. We then have to go on to explain further to get the second mark. What we have to do is we have to put the hydroxyl and the hydrogen groups on carbon 1 are inverted. So it's important that you use these specific terms. And as we said earlier, it allows the easier formation of glycosidic bonds, but you don't have to put that. Now the next question is starch molecules and cellulose molecules have different functions in plant cells. So highlight plant cells. Each molecule is adapted for its function. explain one way in which starch molecules are adapted for their function in plant cells. Now, this is a pretty easy one because you don't actually have to write a lot. So you would just write starch molecules are helical/coiled, so are very compact. You don't really have to go into much more detail. The mark schemes are quite brief with these kind of questions. And actually we do have a last question. Explain how cellulose molecules are adapted for their function in plant cells. Now this question comes up a lot and it is always 3 marks. And what they like to do in AQA is they like to give you a lot of space to answer the question, even though you don't actually need to write a lot at all. So as we said earlier, cellulose chains are long and straight, which are linked by hydrogen bonds. That's something they like you to write. This forms fibres called fibrils, which provide strength so cellulose can support the cell wall. So if you look at the mark scheme from these last 3 questions, so for this question here, the mark scheme says starch is formed from alpha glucose, but cellulose is formed from beta glucose, so that is 1 mark. And the position of hydrogen and hydroxyl groups on carbon 1 atom are inverted, so that's the 2nd mark. Here's the next thing, which is explaining the properties of starch, is we wrote helical and compact, so that it is 2 marks for us. Now you can also write, as I said earlier, starch is insoluble, so it doesn't affect the water potential, or you can write starch is actually large, so it cannot leave the cell. Now the important thing to recognise here is these or, which is written in bold. That means that you have to use the 2 paired terms together, so you wouldn't get any marks if you put it's insoluble, so it can't leave the cell. You wouldn't get the mark if you wrote that, so you have to write what the pairs are in, so insoluble or doesn't affect water potential. You have to do that to get the mark. Now, onto the cellulose question. Long and straight chains, when linked by many hydrogen bonds, to form fibrils. An important point that it's good to know is that hydrogen bonds are actually quite weak, but collectively they are very strong. You don't have to write the word many, but I advise you do. And the last point, it provides strength to the cell wall, as you learned to GCSE. Right, that is the end of the video and all you need to know about carbohydrates for A Level Biology AQA. So let me know if you have any comments or particular requests for videos, and thanks for watching.